Bone substitute materials, BSMs, are intended for use in filling bone voids or defects of the extremities and pelvis that are not intrinsic to the stability of the bone. Zimmer Knee Creations AccuFill BSM is used to fill subchondral bone defects in the subchondroplasty procedure. AccuFill BSM's base material is formed through a patented manufacturing process of amalgamating two distinct forms of calcium phosphate. These include amorphous calcium phosphate and dicalcium phosphate dihydrate, designed to closely mimic the inorganic matrix component of human bone. The result is a material with a unique chemical formulation with embedded metal ions that is comparable to that of human bone. When mixed with a hydrant and implanted into the subchondral bone defect, the material undergoes an endothermic reaction and begins the crystallization process, which consists of two major events: nucleation and crystal growth. The metal ions embedded within the chemical formulation play a pivotal role in maintaining the nanoscale crystals that are similar in size to those in human bone. These differentiating features make AccuFill BSM a 100% bone mimetic bioactive, fully compatible with the human physiology and able to undergo cell-mediated remodeling, mimicking the natural process of bone formation. Once hydrated and in an injectable form, AccuFill BSM has unique fluid dynamic characteristics that allow for percutaneous delivery through small needles without phase separation of material from its hydrant. When injected into the defect, the material, with the help of an excipient, interdigitates within the trabecular bone, yet still preserves vascular pathways. Shortly after the BSM hardens, its surface is infiltrated by the body's cells. The process of bone turnover is primarily controlled by the balance of bone resorption and bone formation. The bone-resorbing osteoclasts form resorption bays. These multinucleated cells form a specialized ruffled cell membrane, which touches the surface of the BSM and increases the surface area interface for bone resorption. Lytic enzymes, when secreted, activate hydrolysis, dissolving the organic and inorganic components of the material. Signals resulting from initial trauma trigger osteoprogenitor stem cells to differentiate into mature osteoblasts. Concomitantly, these bone-forming osteoblasts secrete type 1 collagen and other non-collagenous proteins of the bone matrix, which form osteoid or unmineralized matrices. They also regulate the mineralization of bone matrix. The active matured osteoblasts often get embedded within their secreted matrix and differentiate into sensory cells termed osteocytes. This concomitant process of resorption and bone formation constitutes remodeling. The unique chemical composition and nanocrystalline structure of AccuFill BSM stimulates cell-mediated remodeling. Studies conducted with canine and rabbit subjects accurately depict the remodeling capabilities of AccuFill BSM. This remodeling process helps in bringing back the microscopic structure of bone as the bone heals.